Hopefully everybody's clear on what's happening on Wednesday. Yes? Good. What is happening on Wednesday? Exam, 50 questions, multiple choice. Bring the blue Scantrons. I forget the number, but I posted them on D2L. Those of you who are taking the exam in the testing center, send me an email to remind me, because otherwise I am likely to forget, and you may show up there won't be an exam for you. So, question. Is this review being recorded? Yes, that's why it took me so long to get started. Yes, and I will post it ASAP on YouTube, again, assuming that my software talks to Google software and all these other kinds of things. We shall see. I'll do my best. Other questions about review, what's going to be on the exam, everything? Yes. All of the stuff I'm talking about today is slides which I used before. So there's nothing new at all today whatsoever. Yeah, Stretch?
Just to make sure, I think you said before that any slides, like at the end of the lecture, you do some of the slides in the chapter, those aren't covered, right?
Yeah, so the question is stuff at the end of some of the lectures, if I didn't get to them and didn't talk about them, they're not going to be on the exam. So there were a couple things particularly having to do with the human genome, some of that genomic stuff that we went over, just as skip through all those slides. There won't be questions on those on the exams. And the best way to know what those are is to look at the YouTube, and you'll see click, 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 going through all of them really fast. Okay, other questions? I did post all of the clicker questions so far on D2L. Put them all together into one file. Those are there. For the answers, you can listen to the recordings, or if somebody's really nice, they'll go through and make all those modifications. And you can buy them a beer afterwards. Maybe I'll have to edit that out of the recording. Okay, any other questions, comments, worries at this point? Yes, there are clicker questions today, so you can get your clickers out. And that will be now. Which RNA polymerase is at every replication fork? RNA polymerase 1, 2, 3, DNA primase, or DNA polymerase 2? Ten. Okay, here we are. Yes, as I heard people mentioning here in the front row, it's about RNA polymerase. It was not a typo in the question. Which RNA polymerase are we talking about here? So RNA polymerase at a replication fork. Yeah, RNA polymerases 1, 2, and 3. We'll talk about those later, but they're all eukaryotic. Which has nothing to do with this particular replication fork. DNA polymerase 2, we haven't talked about yet. Talk about really briefly a little bit later on. It's a bacterial polymerase. But DNA primase, of course, is a RNA polymerase, and it's critical at the replication fork because you have to be making all these lagging strands all the time. So that is the correct answer. So it's a DNA primase here. It's a little confusing, but that's the process there. So our next question is The replication initiator binding protein or proteins in Saccharomyces cerevisiae is DNA A, RPA, ORC, helicase, DNA primase. Fifteen, 10, 5. Okay, first part of this question. What's Saccharomyces cerevisiae in terms of what kind of organism? Eukaryote, exactly. So that's the first thing that you want to get out of this question when you're reading a question like this. Okay, so which of these are eukaryotic proteins? Or maybe better yet, which one is bacterial? DNA A is a bacterial protein, so you can get rid of that immediately. So that can't be right. What's RPA? Single-stranded binding protein. Yes. Helicase and DNA primase are pretty straightforward. What does ORC stand for? Saramon's Warriors. No. The origin recognition complex. What's the origin about that? What do I mean by origin? Origin replication, also replication initiator. So those are two exchangeable terms. So it is the origin recognition complex, ORC, which is a bunch of proteins, which is why I put protein or proteins. If this had been E. coli, what would the answer have been? A. DNA A is correct. Okay, so now, again, the rest of today is going to be review, starting out with information transfer. And again, if people need to get up and leave, just please sit in the back and leave quickly, quietly. But yeah, basically what I wanted to mention here in terms of central dogma, we haven't talked very much about these next steps in the process. And basically after the exam, mostly what we're going to be talking about is going from DNA to RNA, this process of transcription and transcriptional regulation, how that takes place. We'll talk a little bit about translation, which is, of course, going from RNA to protein. We've mostly just sort of now talked about replication, how you make more DNA, but also about the individual components, sort of the chemistry, how these things go together, and what each protein, et cetera, looks like together. This whole environmental influence here, now some of that's the epigenetic regulation, but a lot of it also has to do with exactly what's in any of those given sequences. I've had a couple of people ask me about model organisms. That's partly why I put up that question about Saccharomyces cerevisiae. I would expect you to know that Saccharomyces cerevisiae is a eukaryote, and it's a wonderful eukaryotic model organism. So people study Saccharomyces cerevisiae not because they care about brewing or making bread. They study it because they're interested in thinking about a whole diversity of the way that the individual molecules work. So as you know, it wasn't Louis Pasteur, I think it was François Jacob who said, if it's true for the E. coli, it's also true for the elephant. So that's the whole idea of looking at model organisms, is trying to find, studying in depth, a couple of very specific but then very well spread out in terms of their different places in relationship to us, particularly evolutionary relationships. If you think about Saccharomyces cerevisiae here, very basic processes of molecular biology in eukaryotes. If you look at E. coli, that's going to be what's going on in bacteria, and then hopefully, true for the E. coli, the elephant, the Saccharomyces cerevisiae, and the human. Yeah?
On that topic, in your 09 midterm, as far as wanting to study fundamental processes of molecular biology, you had E. coli and E.
So the question here is about a very specific question on an old midterm. Ask me those online and I'll answer those directly. I don't want to go into individual questions on individual exams at this point. Sorry, Travis.
So then are humans something you would study like a model organism?
You could talk about them as being model organisms. And in the textbook, they talk about humans as being a nice model organism because you can ask them questions about how they feel. And you can't, I don't think you can ask any of, if any of you know how to ask any of these organisms questions and get a nice answer, let me know. Or we'll commit you. So speaking of committing, this seems to be one of the subjects that I have the most trouble explaining because people seem to have trouble with it. I wanted to go over the whole idea of paralogs and orthologs in a little bit more detail here. So the idea here, of course, is in the long run you end up with gene families. These are lots of sequences of proteins that are similar to each other and then have similar function. How do they get generated? Due to gene duplication and divergence. And this happens, at all kinds of levels of evolution. So this is just an example of tubulin, which is clearly one of the larger gene families which is out there. The first thing that happens here is you have a gene duplication that takes place. Could be a whole genome duplication, could be a straight gene duplication. We'll talk a little bit about how that happens later on towards the end of the course, but okay. You have a gene duplication. This gene duplication generates, right here, this is just a tree of looking at this, two different tubulins. These are what? Are these paralogs or orthologs? These are paralogs. And I also like to think this is a pair of genes which you've made coming off of the first one. It's a little mnemonic for thinking about it. It's a pair of genes which you generated. Those are the paralogs. So each of these now, there are two of them. These can diverge in terms of their function, how they're actually going to be working, because it's one organism. These can actually, it's one of the great things about gene duplication. It allows you to have evolution take place on these things. However, you're also going to have genes that are related to each other in sequence just because you have different species, and that's what's shown here. So now you've had each of the alpha tubulins, which is continuing through, in this case, we've got humans, flies, worms, yeast. Each of these guys are clearly related to each other in sequence, but they're doing the same thing, and they're only related to each other because they're related in terms of their vertical inheritance in new species. So these guys are all orthologs relative to each other. It's the paralogs to the pair of genes which formed in that one organism. So, okay, yeah?
It really helps me to know that the prefix of ortho means in a line.
Yeah, ortho being across from each other. All of you have taken organic chemistry. Yep, directly across from each other. Okay? Oh, orthos, yeah. Well, okay, so organic chemistry. See, it's been far too long. Let's not say how long it was since I took organic chemistry. But I like the pair. So pair of genes that form in one organism, that's going to be your paralogs. Maybe that's an easier way to follow that. Okay. So again, a lot of this is going to be switching gears a lot. These are zooming through, what? I guess we had 8 full lectures since last time. So just again, picking and choosing the individual bits here. But basically, the important point from this slide is that I wanted to mention that macromolecules, and so I'm talking about macromolecules a lot. That's all of these guys over here, the polysaccharides, lipids, membranes. But as far as this course is concerned, it's really the proteins and nucleic acids that we're going to be talking about in much more detail. All of these are put together, the proteins from individual amino acids. These are your monomers. Those monomers get assembled into multimers. How does that happen? What's the chemical reaction that you do to do that? Yeah, so these are condensation reactions. So it's all about the water reaction. Same thing is true of the nucleotides, these individual monomers that get put together into the multimers. There are few exceptions to that, and again, most of those are going to be these small molecules, things like ATP, that are important for energy input. I feel I didn't do a terribly good job talking about chemical equilibrium, and again, maybe that's because it's been too long since I took Gen Chem. But the idea here, and again, most important as far as molecular biology is concerned, is that a chemical equilibrium here is what happens at the very end of your reaction. If we were all at chemical equilibrium, we'd be dead. So it's disequilibrium, and some people talk about using this in the search for life on other planets, as you're trying to find chemical disequilibria, which is an interesting way of thinking about things. But here, the reason this is useful is it's useful in terms of thinking about the energetics, thinking about how you have to be putting energy into a system and how energy flows through a system. So at this, non-energy consuming state, we have an equilibrium constant, and that's relative to concentration of products divided by the concentration of reactants at equilibrium. And that's the real key point to this, is if you change the concentration of one of these things, one of the other ones has to change in order to be at equilibrium. And the way that changes is just due to the relative rates of each of these. And so you can do these calculations, forward rate, reverse rate, and it's just the ratio of those, which is your equilibrium constant. That doesn't change. Everything else can, but then in order to be at equilibrium, you've got to be able to change those other things. And this is really useful for us in a molecular biology sense, and we'll talk much more about this later, is when we think about binding. And binding you can just think about as a very, very simple chemical reaction where you have a ligand, and we can ignore this little red thing here and the gray one. Let's just look at the blue and the green here. So blue is your protein, green is the ligand. These guys can either be separate ligand, separate protein, or both of them bound together. So your so-called dissociation constant, which is basically just like that equilibrium constant, is going to be the ratio of the two reactants in this case, because it's your ligand and your protein, relative to the concentration of the product, which is your ligand plus protein when they're bound together. And you can also look at this in terms of relative rates. The off rate would be how you've gone from your complex to each of the individual ones. On rate, going back in the opposite direction. As far as we're concerned, and we can do the math in office hours after class, which I will have office hours after class today. We can also try and schedule some tomorrow if you want to do that. We can go through the math, which gives you that the free concentration of your ligand when half of your protein is bound is going to be your KD. Millimolar KDs are weak interactions. Nanomolar KDs are strong interactions. And we'll talk about all of these particularly when we talk about more of the protein-DNA interactions a little bit later on. Yeah?
Can you say that entire sentence over again?
So this last sentence, I'm sorry, which sentence? But
When you said KD, was the free ligand?
right. So free ligand when half of your protein is bound. So half the concentration would be here, half the concentration would be there, and that's what your ligand concentration is at the KD. And again, we can do the math afterwards in office hours if people are interested in looking at that. So, but what that means is if you have a millimolar KD, and so that's the non-bound ligand, millimolar KD means you've got a millimolar of whatever that ligand is floating around not bound to protein. That's relatively weak binding. On the other hand, if you have nanomolar, this would be 3 orders of magnitude difference of that free ligand floating around. That means that almost all of it's bound, right? Because nanomolar is a pretty small amount. So that means it's a very tight binding that you have between the two. It's a slightly different way of looking at it. Yeah?
I assume on the exam
Sorry.
you're going to have some kind of question with numbers related to this. Will we need a calculator?
Okay, so the question is, will you need a calculator for the exam? No, you will not need a calculator for the exam because probably most of your calculators look a little bit like this, which could also have all the notes from the class on it. So no, you do not need a calculator. In fact, you're not allowed to use a calculator. But you wouldn't need one. If you can't do the math that's on the exam or will be on the exam because it's not done yet, you shouldn't be here. No. And if you really do need a calculator, you can come and find me. I will have a calculator, and it'll be a calculator. So this is sort of a continuation of this whole energetics business. We're not going to talk about how photosynthesis works. We're not going to talk about glycolysis. There's that whole section, I think it's like chapter 2 on glycolysis. No, that's not going to be stuff which is on the exam. But the message here is really it's all about the water and then this CO2 into some kind of organic molecule, and those organic molecules are going to be the monomers. And it's all about how these individual monomers go to the polymers, which is this useful chemical bond energy. That's the process that we're really talking about in this class, because it's that polymerization process, how you're going about doing it. Among other things, talking about proteins, and again, we'll talk more about proteins a little bit later on. But I did want to review this because I feel, again, I didn't do a terribly good job of explaining it when we went through it in the first place. So down here at the bottom is just a very short 4-amino acid. People would call this a peptide, usually about 100 amino acids before you start talking about it as a protein, which has an amino end on it and a carboxy end on it, just like each of the monomers, which have amino ends to them and carboxy ends to them. One of the things that I also wanted to mention here is that this whole, basically the horizontal line in here, this is what people talk about when they talk about a backbone. And so when I talk about backbone interactions in terms of secondary structures, it's going to be the carboxyl groups here, the hydrogen groups here. And so I talk about hydrogen bond interactions in the backbone that you're getting for beta sheets and for alpha helices. It's going to be interactions between these kinds of atoms which are here. Side chains, that's what's coming off here in the middle of each individual amino acid. These are the ones that could have a charge to them. Which ones have we talked about that have charges? Arginines, lysines. Tyrosines don't have charges to them at normal neutral. What about this one? It's got a negative charge on it. What would that be? Okay, that turns out it's an aspartic acid and glutamic acid. And we will be talking much more about those. Remember, those are the acidic basic ones. No, I don't expect you to remember all the side chains. Yes, I do expect you to remember aspartate, glutamate, lysine and arginine, but also tyrosine, because we've talked about that quite a bit as well. So again, it's the stuff that we've talked about. I don't remember you to look isoleucine. No, don't bother. So that's our peptide chain. The primary amino acid structure is just going to be the sequence of these guys from the N-terminus to the C-terminus. So primary, secondary is what? Yeah, more specifically, alpha sheets and beta, alpha sheets, exactly, alpha sheets and beta helices. No, although there are some beta helices which are really cool, but that's a different story. So as far as we're concerned, it's going to be the alpha helix and the beta sheet. There are other secondary structures which are there, and that's all about those backbone atoms interacting with each other, not about side chains. As soon as you talk about tertiary interactions, that's where you have side chain interactions happening. That's where you're getting your three-dimensional structure. And once you have structure, you have. But you also have some cases where unstructured proteins also have function. And when we say unstructured proteins, it's not that they don't have any structure whatsoever, it's they have lots of different structures. So this is partly from Eric Iverson's lecture. I just wanted to go over this in a little bit more detail, partly because I just saw a really cool paper where they talked about this second case right here where phosphorylation gives you a really interesting structure. But the idea here, and this is what all these red squiggles are supposed to represent, is that in many cases, some of these proteins don't have a very specific structure. And that's important for their function. It's either important that they have lots of different structures, which would be, in this case over here, where you've got D, basically blocking some kind of diffusion. This is very true in the nuclear pore complex, things going in and out of the nucleus. These guys want to have lots of different conformations. You can think about it as sort of an oily droplets, or lots of oily droplets that are stopping things coming in and out. And it's much better for those to be flopping around with lots of different structures than having one individual structure. On the other hand, it might be really good to have a protein that doesn't have function in some cases, but when it gets into the right signaling milieu, whatever else is going on, then it does have a specific structure, and then it has a particular function. And this allows you then to switch basically something on and off really easily. And again, this is the nice example of phosphorylation, but you could also have binding. So this protein has no activity, no specific structure, unless this protein is around, in which case they bind together, nice structure, gives you a particular function. Same thing is true with this. Here, if you have a ligand interaction, could be one domain of the protein is unstructured, only becomes structured when you have another interaction. And that brings me to structure function. And again, this is hopefully something that Eric talked about, but he will have talked about it slightly differently than I did. By the way, I am writing the questions on this section, not him. Yeah?
Just going back to that previous slide, for tethering, would that be tethering an unstructured protein, tethering two separate proteins together, or an unstructured protein bound to another protein and then binding to like some kind of ligand?
Okay, so the question here is basically, what is C supposed to represent? And I'm paraphrasing here a little bit. Is red supposed to be a separate protein or a piece of that other protein, so like a domain? I would interpret this as this being a domain of that protein, but I think the idea here is it should be much more general. It could be both, but the way it's drawn here, I would assume that we've got a structured domain to your protein and an unstructured domain in your protein.
And it would be bringing two separate or two separated domains together.
Right, two separate domains together, but those two domains are still part of one polypeptide chain, so it's going to be one connected polypeptide. Yeah?
They call it unstructured, but do we have a secondary structure?
So unstructured here really usually is going to mean that you have no defined tertiary structure. There may be some secondary structure there. That's really kind of up in the air. Some cases you will have some cases you won't. Yeah, in the back.
Is the blue blob in C, is that necessarily another polypeptide, or could it be some other micronutrient, vitamin, mineral, or something like that?
Yeah, again, the whole idea here with C is it's supposed to be general. So the answer is yes. It could be all of the above. This could be histone tails, it could be a phosphorylation, it could be binding to a nucleotide triphosphate. Let me get to the next slide. So yeah, that's the idea, is that it could be any of those different things. And it's just a way of getting something which is otherwise not having a stable tertiary structure, and then usually then not having a particular function. And so that gets to this example of the GTPases. These are proteins, again, just represented by blobs down here, that have a particular activity, and that activity is dependent on a particular structure. Well, with the GTPases, which are very well understood, there's a change in that structure that happens. In fact, there are two changes in that structure that happens. The first change is when GTP bound gets hydrolyzed to GDP. And so that's the first change, and usually this now goes from an active state to an inactive state, and usually that's in terms of what it's interacting with. So it's a surface here which changes. That particular surface here is the switch helix which moves. One of the very first ones of these to be studied is the RAS protein, which is mutated in something like 50% of human solid tumors. That's partly why people were really interested in it. And so it's that difference here in the structure here in terms of what, in the case of cancer, leads to too much activation. But in the normal case, this will be active when GTP is bound, inactive because there's a change in structure when GDP is bound. So that's the first change in structure. The second change in structure is when GDP comes off and that gives you another inactive state. So you've got 3 different structures here. One of the big differences between GTPases and what I didn't put up the slide, but certainly, hopefully, Eric talked about as well here, are the kinases. We actually have a covalent change to your protein. And that covalent change is phosphorylation, happens on OHs. What's our favorite side chain which has an OH on it? Tyrosine. And there's also serines and threonines. But tyrosines, any OH on the side of your protein can have a phosphate attached to it. That's a covalent interaction. That covalent bond is much, much, much more stable. Basically, once it's on, it stays on. On the other hand, these GTPases, once you have GTP bound, there will be hydrolysis that happens. And it's a relatively rapid case. You can modify the rate of this hydrolysis, but it happens. And so basically, you're on, but you're only on for a little while. And then you're off again. So I like to think of this as a timing process. You turn on the process by GTP binding, and this should really be a loop here. And then after some period of time, it gets turned off. On the other hand, a kinase reaction, it's like a switch. You turn it on, it's on. You've got to come back and turn it off later. And so these are 2 very important differences in terms of usually signaling, which is critical for everything that will be talked about in cell biology, but also for a number of things, particularly in terms of translation. We talk about this in molecular biology a little bit later on. So switches versus timers, and change in structure, which is leading to a change in function. The other thing that, again, hopefully Eric talked about are this ubiquitination process. Ubiquitin, which is this little question mark up here, is a peptide. It's actually relatively big. Ubiquitin, it's basically the same in pretty much all eukaryotic organisms. Very slight changes here. But again, it's a covalent modification. So in many ways, it's actually pretty similar to phosphorylation. So you put on a ubiquitin. How do you put on that ubiquitin? You have a very general set of proteins, the E1 and E2 proteins. These are involved in getting ubiquitin ready to put onto the green protein that has to have ubiquitin added to it. How do you know which one to put it onto? That's where this E3 protein comes in. This is the one which is specific to the protein that you're trying to bind to, and this is the research that Dr. Singer is doing down in the South Waterfront, also in the biology department here. So he's looking at this E3 protein and a number of different E3 proteins and what they interact with. So they interact with here, and they basically bring this protein that needs to be modified to one of these E2 proteins. We have this covalent change, and that covalent change is going to lead to a change in the structure of that original protein. Could lead to degradation. That's by no means the only thing that you get ubiquitination happening for. Where else do you see ubiquitination that we've talked about in this class? Histones. Histones also get ubiquitinated. In fact, that was the very first ubiquitination to be found, was on histones. You don't want to be degrading all your histones all the time. And so that was, in fact, it's a signaling molecule, actually not that different to what happens with phosphorylation. Okay, other questions on ubiquitination modifications? Yes? What does ubiquitination signal for on histones? What does any modification signal for on histones? So it's an interaction with something else. And that interaction with something else could be your heterochromatin formation, could be euchromatin formation, a whole bunch of different things. It turns out that the main ubiquitination you find is on histone H1. And quite why you get that? Probably involved in opening up your 30 nanometer filament. But you hear me say probably and not have a big degree of certainty in my voice, because it's, as far as I know, not completely clear why that's actually happening there. Yeah?
So you said that it's very similar to phosphorylation. And is that an example of the histone tails? Like when you phosphorylate a histone tail, it has a similar function as when you ubiquitinate it?
Okay, so maybe I misspoke here when I said very similar to phosphorylation. I mean in a very general sense that ubiquitination is similar to phosphorylation because it's a covalent modification. It's not so much as a very specific, because a lot of cases, in fact this case, the polyubiquitination that happens here, is important for degradation of proteins. I don't know of any cases where phosphorylation per se leads to degradation. So it's just the process itself, a covalent modification, which is similar between phosphorylation and ubiquitination. Yeah? So yeah, so the E3 itself is responsible for the specificity. So it's which of these green proteins gets ubiquitin added to it. So it's basically providing that connection between the two. Yeah?
And in this example, once the polyubiquitin gene has been added, at that point the protein would be degraded?
So in this particular case, yes, polyubiquitin has been added. In this particular case, that's leading to degradation. But you can also have polyubiquitin that doesn't necessarily lead to degradation. So there's, again, ubiquitin is used for a whole bunch of different things. Unfortunately, it usually gets pigeonholed into it's just degradation. It's not true. Okay, let's get rid of those silly proteins, talk about interesting molecules like nucleic acids. Just again as a reminder here, review from what Eric talked about, the transforming material here, it's just all about what changes a bacterium and what changes that bacterium even if it's dead. Now this only works for bacteria which are naturally competent, i.e. they can pick up DNA from the environment. And then the figure down here at the bottom is actually not from Griffith's experiment at all. It's actually from Avery's experiment, which is the next one here. But again, the idea here is you add something to your bacteria, that bacteria changes. How did Griffith know? Well, that's because he injected the poor little mice and they died or didn't die, as the case may be, if they'd become transformed. I like the transformation actually here much better in Avery's case because you've transformed cells that grow like this, little tiny things, to these ones that have this big capsid production, or capsule I should say, polysaccharide production on the outside. And so we now know that what happens here is it's the gene for that capsule production which is what got transformed. That's the transforming principle. And that turns out to be DNA, and that's basically what Avery showed in this paper, was it couldn't really be anything else than that. So now let's look at these nucleotides in more detail. Hopefully 3' and 5' is something you're thinking about in your sleep now. The idea here is it's all about the ribose, which is the backbone of DNA so far, but also RNA as well. Here 2' deoxy, that means you've only got hydrogen atoms here and no OHs here. 3' OH and 5' phosphate. So here's your phosphate. Normally, if you're thinking about the monomers that are being added to any of your DNA polymerization reactions, those are triphosphates. So you have 2 more over here on this side. Again, 5' end versus the 3' end. Then you also have at the 1' position of your ribose, that's where the base is attached. And this could be C's, A's, T's, or G's. What's the problem with this particular base? What's missing? 2 things are missing. 2 hydrogen atoms. There should be one right here and right here, which is actually labeled over here. Why they didn't put it in both of them, I have absolutely no idea. But that's the kind of thing that I would expect you to know for an exam, is that those things are missing. There's a problem, clearly, with these things. They're assumed, but if they're assumed there, why are you drawing them out here and here and here? Yeah, so it's assumed here, it's assumed there. Well, but then why isn't it assumed up here? Why isn't it assumed here? Yeah. So again, why you show it here and not there? Again, so that, but I would expect you to include those things. Yeah. So I would expect you, if you're going to draw a base pair, which you're not, but if I were to give you an exam question that said, what's wrong with this? You would say it's missing a hydrogen there and it's missing a hydrogen there. Yes? No? Yeah. No, yeah, it's not just for, well, here, this guanine is fine. But if there were a guanine here, and in fact in some of the other slides you have the same problem. Yeah, no, it's not, it wouldn't just be adenine, but it won't be hypoxanthine either. Okay, more questions about bases and base pairing. Here's another thing in terms of, now, clearly, here we're missing a whole bunch of different stuff in our base pairs, right? So why is it not including all of our, hydrogen atoms here? Why we don't even have the double bonds here in each of these? So oversimplification. I'm not a big fan of it. But what I want to show here is really that you've got anti-parallel. And so this actually is really nice, and it does show nicely that your 3' end here is pairing with your 5' end over here. And you always have opposite orientation of the two strands, and that was one of the two really big things that came out of Watson and Crick's model, is anti-parallel, which they just had to come up with model building. You can't get that from any of the data which were available. Anti-parallel is only because they put the models together. And then the hydrogen bonds as well. Most people just think about the hydrogen bonds, but anti-parallel was just as critical in terms of being able to fit together the structure, and that you end up with a major groove and minor groove. So here's the minor groove down here at the bottom. Here's the major groove here. A couple of things you can see. This one's a lot bigger than this one over here. There's a lot more chemical information, all these blobs here, than there is here. That's just accessible. You can get to it. So if you're thinking about specific DNA interactions, usually with proteins, it's going to be happening most of the time here in the major groove, because that's where you can access the information. That's where the information is available. Okay, questions about DNA structure. Talk about chromosome structure. This is the slide that, for no apparent reason, completely disappeared the first time when I presented it. Just talking about chromosome translocation and ways that you can see that. Here is just a piece of this particular chromosome, which is moved over to another chromosome. And that's classic kind of thing that happens in a lot of cancers. In this particular case, Philadelphia chromosome, completely treatable with drugs that will work against that particular protein, which is made CBR-able. Yes?
Can you go back to the Watson and Crick thing?
Yeah.
Somebody was saying that Watson and Crick thought that everything had 3 hydrogen bonds, and that's how you knew, for example, on the fall 2012 test that the AT pair was wrong.
If we're talking about individual test questions, let's cover that offline.
So, but back to the Watson and Crick, they
Yeah.
thought that everything had 3 hydrogen bonds, GCAT, right?
Yes, which in fact you can get pretty close to a hydrogen bond right there.
That's okay.
Yeah.
You had made a comment that there's one specific thing that fits in the minor groove.
I don't think I ever made a comment about one specific thing fitting in the minor groove. Major groove, alpha helices fit great into. Minor groove, there aren't very many things that fit well into the minor groove, other than salts, which is, again, a totally different story. Take Biochem 492. Tell you all about that. Okay, so again, chromosomes. What are chromosomes made of? Mostly nucleosomes. This nucleosome slide is mostly to show, again, hopefully this is beating the dead horse here. The greens and blues here are histone H3, histone H4. You've got 2 H4s and 2 H3s here that bind at one end of the nucleosome, H2A, B, H2A, H2B binding here, and then all the tails. So you can nicely see one of these tails hanging out here. This is the histone H3 tail, which of course is the one which is getting heavily modified. In the back, yes?
Just to verify, you did say that the lysines and arginines connect with the minor groove, the DNA and the histones, right? That's the thing?
They can. It's not always the case that that's going on. Well, one of the things to go, if you look at this nucleosome structure, many of the interactions which are happening here, and here's a really nice example. There's your minor groove right there. That is interacting directly with the nucleus. And the major groove is out here on the outside. So lots of interactions are in the minor groove, but they're not going to be very specific interactions. And so the key there is the specificity. So non-specific interactions, very often it's going to be in the minor groove. The specific interactions are going to be in the major groove. That could be, side chain, could be all kinds of different things. We haven't looked at the scale so much of these things. We will when we talk about DNA-protein interactions a little bit later on. There was another question over here? I think? No? Hopefully answered? Pardon? Okay, good. So the main thing here is, yes, it's these 8 proteins, 8 peptide chains which are here in the core of the nucleosome. But the nucleosome itself, just like all these zomes, is nucleic acid and protein together. Because they're all wrapped around nicely like this. Oh, sorry. Yeah.
You probably just got to go over it, but I was just wondering because H2A and H2B fall off of the electrons. I was just wondering if the DNA stays coiled when they're not bound.
Oh, so the question is the DNA stay together when they're not bound? When you get have H2A and H2B which are exchanging? So the question, okay, what is happening to the DNA? What's happening to the nucleosome when you have H2A and H2B which are exchanging? I'm humming and hawing here because we don't actually have that good data on this, but everything that you look at when you do modeling says there actually really has to be something which is coming apart in that process. And so if you think about it, let's back up to the nucleosome here. You can have this piece of the nucleosome, this wrap part of DNA, that's still clearly associated with H3 and H4. There must be some kind of unwrapping that takes place down here so you can have that exchange that takes place. And that's, again, but we don't, the problem is this is a very dynamic process, the exchange, and so it's very hard to get high resolution imaging for. So, that being said, yes, those can exchange, H2A and H2B, get to in just a second, but the process is not that frequent. They can exchange, but most of the time, your histone octamer actually stays together. It's pretty rare that you have those. You can have exchange, but it's not something which is going on all the time. Yeah. Yeah. Oh, so this is just basically the whole idea here is supposed to show that this basically looks a lot like that, and it's just that you can stack them on top of each other. A nucleosome is really symmetric. And that's all that it really means. The entry and exit could be either way around here. This is to show that those are different in terms of which way around they've been turned. Okay, so getting back to this comment about exchange, is that exchange doesn't happen in the absence of some of these other proteins, the chromatin remodeling complexes, which can be moving the DNA along the nucleosome, or could in fact be dissociating either H2A and H2B and having that exchange, or completely dissociating the whole thing and putting together whole complete new nucleosomes. Histone chaperones are needed, again, mostly to make sure that you have the right ones which are coming together here. Chromatin remodeling complexes all are ATP dependent, so you need ATP to be moving stuff around. So that just basically says that that structure, again, the one that we looked at back here, is a pretty stable one. It's not going to be popping on and off all the time in the absence of actually spending some energy to do it. You do have some breathing, but that's not completely dissociating. It's little pieces which are dissociating as time goes by. Reminder that it's not just the nucleosomes. There are many other proteins which are associated with the DNA. In fact, about the same amount of protein as histones, which are there. There's all different proteins which are bound to your DNA. So we've talked about scaffolding proteins. We've talked about barrier proteins, some of the regulatory proteins. All of these things are associated with chromatin, as well as the nucleosomes and the histones which are there. So again, just a reminder that all this is what's there, and most of your chromatin, most of the time, is going to be in these 30 nanometer filaments. I want to spend a couple of minutes talking about supercoiling because this always seems to be confusing for people. Thanks to whoever prompted me to ask about videos. I did find quite a nice one online, which is posted on D2L now. But basically, if you're looking at a closed circular DNA, or actually even something which is a really long linear chromosome like you have in larger eukaryotes, it's basically a relaxed form where all that you have is twist going around it, which are the two strands wrapping around each other about every 10 nucleotides. So if you have what's called a relaxed DNA here, it's just twist. As soon as you change this in any way, which of course you have to have a topoisomerase to be able to do, you can change either the twist, which is topoisomerase 1, because it's one strand that you're changing. You can change the writhe, which is the two strands passing through each other. That's topoisomerase 2. Either of these guys changes twist or writhe. Those are the linking number. Linking number is just twist plus writhe. And that can give you either something like this with writhe, with double strands crossing over each other, or untwisted. These two are completely equivalent to each other. Untwisted and writhing, or most people talk about this as being supercoiled. I didn't bring my little phone cords with me, but again, in office hours, more than happy to go over that with you. Yeah?
So you said those two are equivalent to each other, but it wouldn't take topoisomerase to switch between them?
No. There's no requirement for topoisomerase here. This side requires topoisomerases. This side does not. These are interchangeable. Which is probably partly why we've got a lot of negative supercoiling in the cell, because that makes unwinding a little easier. Okay. Yeah, sure. Okay, to get to negative supercoiling, yes, you have to have topoisomerases. But once you have negative supercoils, the writhe versus untwisting is an equivalent situation. If you are untwisted, yes. But only if you're untwisted. If you're relaxed, you don't get writhe. As soon as you untwist or you twist across, then, and that's so this actually negative supercoiling is great. Sorry, it's kind of an aside here, but negative supercoiling gives you compaction, but it also helps get those two strands apart. And that's probably why most of the supercoiling in a cell is negative, because it's helping getting those two strands apart. Because you need to do that for replication, you need to do it for our transcription. Yeah.
So the topoisomerases, you said that topoisomerase 1 is required for twisting. And
So topoisomerase 1 is required for twisting, topoisomerase 2 for writhing. It's those two strands across.
So if you were like holding open if you were like holding the one on the right and you let it go, it would like twist around itself.
So if you had, the question here is if you were hanging onto this and then let go, it would go and look like this. And yeah, when you say let go, it means allowing this to twist back together. If you allow this to twist back together, the only way that you can have that be stable is if you get these writhes to happen. So that's what I mean when they're interchangeable. Yeah?
I'm kind of confused with negative and positive supercoiling. How do you get either one? So negative you get from cutting.
Okay, well, so no, let's maybe back up. So the question is difference between negative and positive supercoiling. As far as we're concerned for this class, and there's some details that we don't need to get into now, but positive supercoiling is going to be the same as over-twisting, so taking your DNA and twisting it more, and negative is going to be equivalent to untwisting it and going in the opposite direction. That's the easiest way to think about it. Yeah? the question is, why can't topoisomerase 1 change rise? It does change rise. So yeah, it's just change so both of the topoisomerases, sorry to get the topoisomerases change linking number, and linking number is twist plus rise. And since those are interchangeable with each other, topoisomerase 1 can end up changing your rise, topoisomerase 2 can end up changing your twist. Because they're equivalent, they're interchangeable. Okay, now everyone's completely confused about this. Talk about chromatin modification. Just the main thing here is that if you have one kind of modification that takes place, usually through one of these histone modifying enzymes, here it's just the acetylase and deacetylase. Same thing could be happening with methylase and demethylases. Once you have this change, now you have a code reader protein that is going to lead to that same change happening in that same part of the DNA until you get to some kind of barrier sequence, barrier protein, etc. Usually that's going to be in one particular looped domain of your DNA, and that you can actually visualize by looking at transcriptionally active parts of the chromosomes. They're all lined up in the salivary glands of insects, particularly in this case fruit flies. So if they're compacted here, this will be your heterochromatin. Here's your euchromatin. Clearly a lot more space which is there. How do you know? That's because each of these dark dots here corresponds to the RNA polymerase. So expanded chromatin, it's your active chromatin. It's moving things apart relative to each other. What's in our genome? Again, hopefully we're beating the dead horse here. Protein coding is really minuscule, and viruses are way cooler than anything else. That being said, I did want to cover a couple more things on the human genome that I felt I went over a little bit too fast before. And one of those is, if you look at these exon sequences, again, that 1.5% of our genome that's actually coding for protein, those are incredibly well conserved. And if you look at all these different organisms down here, all the way down to the fugu, the pufferfish, exons are incredibly well conserved. It's because each of these lines here represents that it's a similar sequence. On the other hand, you also have a number of these other things, like these over here, these multi-species conserved sequences. So this block, and this block, they're not as well conserved as you have exons here, but they're pretty well conserved. If you look down through these, particularly some of these really different organisms like the non-placental mammals, the marsupials here, which are well conserved. That's one of the huge questions that we have right now in molecular biology is genomics. It's trying to figure out what those sequences are doing. Because they've been conserved throughout evolution. There's a reason that they've been conserved. What is that reason? And so that's one of the main foci. So we can ignore most of the rest of this stuff. We're going to concentrate on some of those things. So human genome, what's in a human genome? 3,244,463,819 base pairs. That was in September of 2012. Still a bunch of gaps. And so there are a lot of gaps in the sequence. Most of that has to do with telomeres and centromeres. Repeated sequences. Repeated sequences are still a big, huge problem in terms of genome sequencing. So somebody says the human genome, we're actually still missing quite a bit of the human genome. And the human genome was volunteers of different racial backgrounds originally. Well, that's who happened to be in the lab at the time and they could get blood samples from. So one of the things which is happening now is what's called the 1,000 Genomes Project, which is where people have really now tried to get a lot of human diversity and look at all of those different genomes and try and find, in ways not dissimilar to that conservation I just showed in different organisms, in different people, and trying to figure out what each of those different things are that are there. We'll ignore the good guys and the bad guys here. Really cool race, but that's a different story. Just wanted to mention here, I mentioned some of the data from the ENCODE project. This is now going and trying to figure out what each of those extra conserved regions are doing, and even also some of the other bits which are in there. As I mentioned at the end of, I forget which lecture, about 80% of our genome is being actively transcribed, i.e. it's being made from DNA into RNA. Well, why is that? What's all that stuff doing? Since only 1.5% of it is actually coding for protein sequence? So that's a really open question. We have a relatively small number of genes. Again, this is something that goes down every single time. And the other, I think, really amazing process, if you go to this link, this is a human genome. In fact, there's a whole bunch of human genomes, including Craig Venter's genome, Jim Watson's genome. You can go and get these. They're freely available online. Go and look at somebody's genome. It's actually pretty amazing that you can do that. Yeah?
Sorry, just to clarify, you say that 80% of the human genome is protein coded but not being transcribed?
No, 80% of the human genome is being transcribed, but 1.5% of it is protein coding. So what's the other 78.5% being transcribed? It's being transcribed into RNA, yes. So many of those are non-coding sequences. They're repeated sequences. They're who knows what all. And so, again, it's given us a lot of stuff to think about and work on. Yeah?
When you say that only 2,000 protein proteins are unique, does that mean unique to humans?
Unique to humans, yes. Which means there's not much which makes us different from worms and flies and things. Yeah?
On the previous slide, you had this thing between the chicken and the boo-boo that said a 100%, 50% identity. What is percent identity?
Percent identity is the number of nucleotides which are identical in a particular window, and that's what that is if you look at 100 nucleotides, if 50 of those are the same, that's a 50% identity. Yeah? Okay, the question is over 30% identity. I don't mind not finishing these last couple of slides. If you're interested, we can talk about them later. So again, it's not critical. 30% identity is the question. Unfortunately, that's the protein sequence versus nucleotide sequence. So 30% and above amino acid identity is going to give you same structure, which is going to give you the same protein family and usually the same function. And why don't we just leave it at that? Because, same structure, same function. That works really well for me. Best of luck on Wednesday.